Hi, welcome to Neuroverse
A podcast hosted by Clara.
and Carolina, where we discuss all matters from neuroscience to philosophy and beyond.
Today's episode is on measuring the brain in time and space. And we've been wanting to do this episode for quite a while now because as we become more familiar with the world of research and learn more about the tools that are available to probe the brain and to measure brain activity, it's become quite clear that there's always this sort of trade-off between spatial resolution and temporal resolution. And what I mean by this is techniques that have high spatial resolution can tell you exactly which area of the brain is active, whereas techniques that have a high temporal resolution can tell you when the activity occurs. And in general, it seems like the greatest challenge may be to understand the brain's activity in space and time together, because techniques either focus on where the activity is occurring or when the activity is occurring.
Yeah, and I feel like in the beginning of neuroscience research, these two types of techniques were very separate. But now, as we're advancing more and more, more molecular tools which have high spatial resolution now are also having quite a high temporal resolution.
Yeah, and more methods are being formulated that use a combination of techniques together that can give you spatial and temporal readouts.
Yeah, from me personally, I think the papers I like the most are the ones that do have both techniques.
Yeah,
Yeah,
but it's easier said than done.
of course. I also, before we dive into it, I think another inspiration for me for doing this episode is when we were having a conversation with Tact that we've done an episode about science art with, and we were explaining how in neuroscience there's always a trade-off between spatial and temporal resolution. And we realized then how this may not be intuitive for everyone. And so we wanted to explore that further. And I also found it interesting how, from an outsider's perspective, it's hard to understand why you can't do both in neuroscience.
Mm-hmm.
And I think, I really want to dive deep into why is it that with molecular tools or with spatial tools, we can't also have such a high temporal accuracy.
Yeah. Mm-hmm. So to start off with, I want to give a couple of examples of techniques to further kind of show why there is this spatial temporal trade-off. So starting with techniques that look at activity of the whole brain. One commonly used technique is neuroimaging, which I'm sure we're all familiar with. You've probably heard of MRI, magnetic resonance imaging. These techniques can record brain activity with a high spatial resolution because you can visualize where the activity is occurring in the brain. And another thing I also want to explore in this episode is how the understanding spatial dynamics and versus understanding temporal dynamics relates to understanding the functional properties of these mechanisms in the brain.
Mm-hmm.
And so in neuroimaging, for example, we have a specific form called fMRI, functional magnetic resonance imaging. And this utilizes the blood oxygen level dependent or BOLD signal. And that is based on localized changes in brain blood flow that is thought to be an indicator of neuronal activity because blood flow is linked to neuronal activity by neurovascular coupling. But of course, this is very correlative. Yeah.
Yeah, like as we mentioned in the episode on coffee, there were studies that showed that people who drink coffee have slightly less oxygenated brains. And that's something that in an fMRI scan, you can misinterpret the results because oxygen is not the sole measure of neuronal activity.
Yeah, so the effects of caffeine on blood flow must be taken into account when you're looking at neuroimaging because changes in blood flow are not necessarily indicative of changes in neuronal activity in that case.
Exactly.
Yes, and another really interesting thing with neuroimaging is there's also ways to measure brain connectivity through imaging, but the way this is done is by correlating the activity of different brain regions during a task or during resting state as well, and this is taken to be indicative of functional connectivity between brain regions. But again, I think this is really interesting because while it's easy to go ahead and assume, oh, these brain regions are connected, you have to really remember that what you are actually showing is that these two regions are actually co-activated. And that doesn't necessarily mean, and then now we get into the whole structure function debate of whether structure equals function and whether a functional readout like coactivity can be indicative of the structure.
Yeah.
Anyways, another interesting form of imaging is also SPECT, single photon emission computerized tomography, or PET, positron emission tomography. And these use radioactive substances like radio tracers to tag specific proteins to visualize changes in metabolic processes or in chemical composition. And these are good indicators of what is happening at the functional level. For example, this is commonly used to tag a synaptic vesicle protein called SV2A that is expressed at the synapses of neurons. And by tagging this, you can look at the synaptic density across the brain. But ultimately, yes, all of these methods, they kind of lack that temporal precision, and they're more focused on understanding where the activity is happening. And so if we want to look at specifically temporal dynamics, then we turn to electrophysiology.
Yeah.
For example, EEG, or electroencephalogram, where you put electrodes on the scalp surface of a human or any animal, and you can pick up electrical signals. And this can give you millisecond precision, as you can record the activity that's occurring across, seconds to hours.
However, it also has limitations in that it can only record activity from more surface layers of the brain.
And the signals that you're picking up, they travel through layers of neural tissue, cerebrospinal fluid, and bone. And all of these obstructions distort the signals along the way. So it's difficult to firstly estimate the neural source. Although there is a method called magnetic encephalogram, MEG, which it picks up magnetic signals, and so it's better at estimating the neural sources of the signal. But nonetheless, you don't really know what exactly you're recording, like what the electrical activity corresponds to, because it is really just a scalp surface level signal.
Yeah. I know that EEG is primarily used in sleep studies, and it's great if you want to use humans as your model. But yeah, they are quite limited in my opinion.
Yeah.
And I have a preference for invasive electrophysiology techniques.
You want to get down to the core of it.
Yeah, absolutely.
Yeah. But I think this is a good starting point to kind of introduce the concept of spatial methods like neuroimaging focused on the molecular aspect of the brain.
Yeah.
Such as the example with tagging SV2A proteins in PET imaging, whereas electrophysiology is more
about the electricity of the brain.
Yeah, the biophysics. Yeah, so both spatial and temporal methods can be applied across many different scales. And the ones I've talked about so far have been at the grossest level, the largest scale. But yeah, we've got loads of other invasive methods at the smaller circuit or single cell level.
Yeah, so one of our favorite methods is patch clamping. And patch clamping is when, with a pipette, you create a vacuum seal on the neuron, and then you can break into the membrane and record the electrical activity. And you have access to the activity happening in terms of, you can estimate what channels are open or closed based on the phase of the action potential or the EPSP that you're recording, whether it's depolarizing or hyperpolarizing. And you can do whole cell patch clamping, which is when you record the activity of a whole cell. You can also record activity from single ion channels, which is incredibly challenging. And you can do this using brain slices, and you keep the brain slices oxygenated, and so the cells are alive. Or you can do in vivo patch clamping, where you go in with a pipette directly into the brain in a live animal.
Yeah, so this is electrophysiology, but at a single cell level, which is insane, and I love it.
Yeah, it's really cool.
Yeah.
My favorite aspect of it is just the dynamics, the immediate response that you get.
Yeah, so I've had minimal experience doing like all forms of electrophysiology, and I've just, even though they're all giving you a readout of the electrical activity that's happening in real time, it's very, very, very different from my experience, because with So with patch clamping, where you're literally just poking a hole in a neuron and then looking at the behavior of this amazing neuron, you really can tell a lot, like you mentioned, about what's happening molecularly or with the ion channels. You can infer the processes that are occurring in this neuron as it's undergoing an action potential or whatever spontaneous activity even.
Yeah.
Whereas with EEG, it's really hard to tell what's happening when you're just there recording the activity. It's much more of a, after you gather the data, it has to go through pre-processing and cleaning, and then you decide what you want to extract from it. And there are so many things you can extract from it, such as isolating different frequencies of oscillations, but it's a lot less substantial and immediate gratification.
Yeah.
And much more thought has to be put into what does this mean, just because there's so many factors that are contributing to this one signal.
Yeah.
So even though they're both electrophysiology, they give you such different pieces of information.
Yeah. A good in between, I guess, between these two. One is very cellular, like single cell. The other one is more whole brain.
Yeah.
Is silicon probe recordings. So the one I'm most familiar with is neuropixel recordings. So these are really thin shanks that have hundreds of electrodes that can then record hundreds of cells. And it's great because they're thin and long, so you can record from multiple layers of the brain. However, as we've mentioned before, there's very low spatial resolution.
You're only measuring where your one probe is located in the brain. So even if it can go across the layers, in terms of the whole cortical surface, it's only in that one little point of the cortical surface.
Exactly.
So you are restricted. Like, you can't put electrodes, you can't just stick them everywhere throughout the brain and know the activity that's happening everywhere all at once.
Yeah. Although it is still cool, because then you can also see activity like, for example, when this specific layer is active, this specific layer is inhibited, and vice versa. And then maybe you can notice something about the circuitry that you didn't before.
Yeah.
Which is quite cool.
I mean, yes, I think neuropixel probes has the highest density of any probe to measure neural activity. So each probe contains 384 channels, and you can implant 2 4-shank probes, which together has 10,240 recording sites.
Wow.
Yeah, so it's very high density. But then I think it's really interesting that you have to put in a lot of thought as to where you're going to place your probe, because they are this long, kind of, I guess you could say, rectangular shape. So, for example, you could go from the top of the brain straight down and just record like all the layers of one specific type of cortex, like say visual cortex or somatosensory cortex. But you could also put it from the back of the brain and kind of like inject it to the center of the brain, and then you could get the cortex, and you could also get some like subcortical structures.
Yeah, and there's even like chronic neuropixel implants, which I think is crazy because neuropixels are so delicate. But yeah, it's great to look at things like learning over time.
Yeah.
A couple limitations I want to point out with neuropixel recordings, though, is that there's a lot of noise that you also gather. And so oftentimes the data gets filtered through something like kilosort, which is a set up algorithm that scientists have determined specific thresholds of what is considered noise and what isn't. Obviously, this isn't perfect, so sometimes labs have their own preferred method, and I think that this creates a lot of trouble with reproducibility in electrophysiology. And I'd like to quickly mention here that part of the work that I'm doing in SWC is part of the IBL, which is the International Brain Laboratory, and it's this really cool international organization that their main goal is just to have reproducible electrophysiology recordings. And they released this paper across several labs. The mice had to do all the same behavioral experiment. They recorded from the same sites, and they showed that they were able to reproduce the results, which is really exciting. I think this is the first time that this type of reproducibility has been shown.
Yeah, very important, definitely.
Yeah. So even though there's limitations, there is hope for reproducibility and accuracy in science.
Yeah. So now I want to talk a bit about techniques that have made progress in finding a good spatial temporal compromise in a way and trying to provide information about both.
Yeah.
So one that is really widely used nowadays is calcium imaging, and this is basically a form of spatially imaging neural activity that also gives you temporal information.
Yeah.
But is ultimately still limited temporally in relation to, say, electrophysiology, which the readout of electrophysiology is like a voltage signal.
It's immediate. There's no temporal gap.
Yes. And calcium imaging, the method is based off of the concept that when neurons are activated, their intracellular calcium levels increase. And so by tagging calcium with fluorescent probes, we can visualize these changes in calcium over time, and that is taken as a readout for neural activity. This generally seems to work and correspond to neural activity, but calcium signals can also be changed by cell signaling processes that are independent of neuronal firing. And so it may overestimate neural activity. And also, there isn't that fine-grained temporal resolution because the biological mechanisms that alter these ion calcium levels take time. So there is this sort of time lag, and you don't get these sharp voltage responses. Instead, you get these slower fluorescent signals. But nonetheless, it is pretty cool because you can use calcium imaging to measure neuronal activity across large areas of the brain if needed. Also, an advantage of calcium imaging is you can target specific cellular populations using the genetic tools that we have. We can specifically express the fluorescent indicators in, say, like a population of excitatory neurons that we want to focus on. And then you're only reading out the activity of those neurons, which is actually an advantage over electrophysiology. Other than patching, where you're targeting a single cell, the signals in neuropixel probes, for example, are coming from all the neurons that surround the probe. And something that's being used by some labs is called wide field calcium imaging.
Which is super exciting.
Yeah, so how it works is you can image the whole surface of a mouse's brain across like the whole cortical surface. So you can really see what's happening in every single cortical region. Of course, this is just the cortex. I don't think this has been used deeper in the brain.
No, it hasn't, but there's a new 3-photon head-mounted imaging device published recently in 2022. So they wanted to address this issue that usually you only have surface-level activity, but they were able to measure in freely moving mice across all cortical layers.
Yeah, that's really cool. Definitely probably quite expensive.
Yes, definitely.
Because the point with imaging is, so normally when you image, it has to do with how light enters the sample and then is emitted from the sample. And if you have a 2-photon microscope, it basically uses 2 photons, and I'm not going to go into the details, but it can penetrate deeper in the brain. So a 3-photon microscope can penetrate even deeper, and so it makes sense that they would try to use that. Also, something really interesting with calcium imaging is a lot of people use calcium imaging to study the hippocampus, which is a structure pretty much in the center of the brain. And the way they do this is a really neat trick where you put a prism, like a glass prism, adjacent to the hippocampus, and then you've got a lens on top of the prism.
That's
so
cool.
It literally, like, it's like making a microscope in the brain,
Oh
where
my
the
God.
light bounces off of the prism into the hippocampus and then, back up to
That's amazing.
get the signal. Yeah, and that's used for, and it's insane that, I mean, I'm sure it affects the brain tissue, but it doesn't hurt the animals are still fully functioning.
Yeah, it's getting more and more advanced, and the spatial and temporal resolution gap is getting smaller and smaller.
Yeah, with nifty tricks like glass prisms.
And 3-photon microscopes.
Yeah. And then another really important method, arguably the most progressive with the whole spatial temporal compromise, is voltage imaging. And this is where you can use a fluorescent probe that measures changes in the membrane voltage. So whereas calcium imaging is using a biological readout, calcium levels, to indicate activity, voltage indicators actually directly measure the membrane voltage changes, but with that spatial readout of a fluorescent signal.
Amazing. Yeah.
So I thought that voltage imaging was like something really new because it's not really being used that often. Only a few labs do it, and a lot of labs are trying to use it, but it's not that well established yet, and they're still creating better, more accurate voltage indicators using, complex genetics. But as I was preparing for this episode, I actually discovered that this method was conceived in the 1980s.
Oh my God.
Yeah, there's a paper from 1982 where they described the first fluorescent probes to do this.
Yeah.
And it's crazy that it's kind of taken this long to progress because I mean, people are on it, but it's just, I think it's very challenging
Yeah.
to make sure that the probes are doing what you want them to do.
Yeah, whenever I think about neuroscience, within the last 50 years, sometimes it can feel like how much do we really know compared to 50 years ago? But in terms of technologies, like of course we're advancing, like this super cool three photon microscope is something that definitely wasn't around then.
Yeah.
But the core ideas haven't really changed. It's just, yeah, it's just building up on each other, and I feel like the core is still the same. Does that make sense?
Yes. And something I want to mention along the lines of this is that, let's say, the technique that's close to both of our hearts, patch clamping, is like a really ancient technique
Yeah.
in relation to all the others, because it was kind of in the 1950s that Hodgkin and Huxley famously started using this in giant squid axons to study electrical activity, and they kind of modeled the biophysics of single neurons using that. And so there's a lot of talk about, oh, patching's going to die out, you know, and
the robots are taking over.
yeah, the people are making robots that can patch,
patch.
which is crazy because it's very challenging, and it does really require
And
like
intuition.
yeah, it requires kind of that human intuition of just feeling how to get access to that neuron, which is weird to describe if you've never done it before. But
Yeah.
ultimately, it is still always being used, despite like it's so spatially constrained. It's one single neuron, but people are still proving for it to be a valuable way to understand how the brain works.
Yeah. And something which I think is really valuable in patching that you don't have in like silicon probe recordings or EEGs, you have subthreshold and suprathreshold activity.
Yeah, so
Within-
action potentials are above the threshold, but then subthreshold activity is still very informative as to how neurons behave. And also, neuroscience maybe was more action potential focused beforehand. And now people are realizing there's so much subthreshold activity that influences everything.
I just wanted to say one more thing. So, in preparation from this episode, I actually looked at the introduction of the annotated guide of the Hodgkin Huxley book. It's by Indira Raman and David Forster. I really recommend. And they say this thing where, I'm going to paraphrase, but basically, imaging methods permit neuronal activity to be tracked through non-electric means. And despite the fact that these are powerful ones, and the visual domain being the native language of humans, images of electrical signals can still be limited. And they compare it to translations of poetry, in that the sense is there, but the nuance is often lost. And that you need to enter the brain's own language, and that without entering the brain's language and only having a proxy of a translator's dictionary, the represented calcium signals, metabolic products, and gene expressions, in their opinion, don't match the accuracy of the electrical readout. And I think that plays a lot into both Clara's and I's bias, and that we do have a slight preference for electrophysiology techniques. And I think this sums it up quite beautifully. But I'm really excited about like future imaging techniques that merge the two worlds together.
Yeah, I think it's also important that they mentioned the visual domain being the native language of humans, because I have to admit I'm very biased to temporal measures, and I was reflecting before this episode on why, and I think it's because I am aware that there's a human bias towards spatial measures because we can visualize it more easily. Like, you know, looking at a single voltage trace is generally not as exciting as looking at a bright, colorful picture of all these lit up neurons. And I do love the excitement of looking at those beautiful pictures. But I think with temporal measures, I feel you can tell more about the dynamics. There's more of like an enigmatic message there. But with the spatial measures, the dynamics that you see, I feel like they're more correlative. Like you can look at where this activity is, oh, that correlates, that correlates. But then with the temporal dynamics, it's like another layer that I think we as humans tend to think in less. It's like another dimension that seems to me a bit more complex. I think that's why I'm generally drawn to it. But you definitely need both.
Yeah, absolutely.
So we're going to sum up this episode here. If you enjoyed today's episode, the best way to support our podcast is by sharing it with your friends and family and leaving a review on our Spotify page. You can also check out our website for any resources and follow us on social media on our Twitter and Instagram at Neuroverse_pod.
Thank you for listening.